Synergistic effects of nitrogen-containing triterpene saponin is a very exciting and interesting mode of action. The first nitrogen-containing triterpene saponins, including amino-acyl moiety linked to sugar chain, called as saponin 1 from Gypsophila trichotoma roots, and study results show that purified these saponins had the significant ability to enhance the cytotoxicity of target toxin compared to other non-nitrogen containing triterpene saponins. The study results show cytotoxicity on the EGFR transfected knee cells was analyzed both in presence or absence of saponin 1, and the results interestingly show that the combination with amino-acyl saponins in non-toxic concentration show that the saponins cause the toxicity increase in some tumor cells. Notably, Gypsophila saponins is also called goat cap saponins because of the first sugar moiety is glucuronic acids is generally, and then this Gypsophila saponins is also known as goat cap saponins, have been proved as enhancer in anti-tumor target in gene therapy. My presentation includes two sections. First one is nitrogen-containing triterpene saponins, and second one is sulfur-containing triterpene or steroidal saponins. Here we are in second section. Sulfur-containing saponins are known in sulfur groups attached, sometimes sugar parts, sometimes carbon skeletal, or carbon skeleton is also known sapogenin or Anglican section. In here, we can see the sulfur groups attached to sugar parts. The first mode of action of sulfur-containing saponins is signal transduction anti-angiogenesis. Tian and colleagues examined the anti-angiogenesis and anti-tumor activities of a sulfur-containing saponin compounds purified from a sea cucumber. Inhibition of angiogenesis was assessed using proliferation, migration, adhesion, tube formation, apoptosis pathways, and PE-treated some human cells. The in vivo chemassays were used to assess inhibition effects of sulfur-containing saponin molecules on physiological angiogenesis way, and immunofluorescent microscopy was used to examine tumor microvessel density and apoptosis in sulfur-containing saponin molecules treated mouse in some tumor cells, and the results show that sulfur-containing saponins affected the signal transduction and showed anti-angiogenesis property. Another mode of action or physiological actions of sulfur-containing saponins is enzyme inhibitory activity. Frondosid A from C. Cucumberia frondosa, which is a sulfate saponin or sulfur-containing saponin molecules, was shown to suppress the serine choline protein kinase, abbreviate PEG1, dependent growth of lung cancer cell lines, as well as pancreatic cancer cells. And these saponin molecules is the first marine compound that selectively inhibits this serine choline protein kinase in vitro conditions. Another pharmacological properties of sulfur-containing saponins. A current study has demonstrated that the radioprotective activity of new immunomodulatory lead material, cumazin, a complex of monosulfide triterpene glycoside from the edible C. Cucumberia japonica, and the results show that sulfur-containing saponin molecules had a radioprotective activity in particularly cobalt isomers. The pharmacological properties of sulfur-containing saponins, the last example is immunomodulating activity. And immunomodulating activity is also important for some physiopathological disease. And saponins of a holostan series, holotrin A and B, holotrin A2, and holotoxin A1, and Dammaran series, which is a type of sapogenin groups or a type of Anglican groups, was investigated by the research team. In low amount, the triterpene glycosides demonstrate mitogenic activity and regulated the immune system. The similarity in the activity of glycosides was first all observed with respect to dose-dependent manner of their effects. The diametrically opposed action of the high and low concentration. The expression of the effects was probably determined by the chemical structure of the triterpene glycosides. Liberation of soluble mediators served as a secondary signal to clonal expansion and differentiation of the cells. Conclusion. We can say that nitrogen and sulfur-containing saponin compounds may be relatively rare in nature, but they have fascinating and promising pharmacological properties. We know all the saponin compounds have some biological properties, including cholesterol lowering action, antibacterial action, antiviral action, hemolytic action, and other biological actions. But in particularly, nitrogen and sulfur containing compounds show the anti-cancer and membrane receptor proteins regulatory actions, including maybe enzyme and maybe other receptor proteins. Thank you very much for your attention, and you see my master students in our department. And I would like to thank my master students and of course the symposium organization members, and see you next time.